drug industry and how they fix using molecular medicine. So molecular medicine is a branch of medicine that helps you to diagnose and treat multiple diseases by understanding the way genes and proteins act. It uses many techniques such as restriction of enzymes, DNA hybridization, rapid DNA sequencing, and genetic engineering. I'm going to be focusing on genetic engineering and rapid DNA sequencing. So, drugs can cause multiple problems, and one of them are genetic diseases. So genetic diseases are caused by drugs by altering the gene expression and the gene function in a person's life. And this causes a mutation within the genes which can spread from cell replication. Therefore, genetic diseases can be inherited from parents and it will be passed on. What I recommend for a solution is the use of viruses to deliver new genes to affect the affected genes, to cure the affected genes. If viruses can cause the diseases, then they can also be the cure. Viruses contain genetic information and protein codes. However, viruses cannot replicate on their own. Instead, they must infect a host cell and replicate themselves from their machinery. They stick to the host cell and release the genetic information. So if they spread the genetic information, then it will spread amongst the body. But if we can modify the genetic information inside the virus and cause it to be like a cure for the disease and allow it to spread, it will just cure the disease inside the body, and this is why it's used as vectors to deliver the therapeutic gene and replace the mutated gene. So the mutated gene would go away, and then the therapeutic gene inside the virus would fix it. And then other problems such as birth defects. When a mother has drug intake during birth, it can cause heart defects, lower than normal, and multiple other birth defects. How we can fix that is the use of genetic engineering in the embryo by using this technique. And this method.
What is CRISPR?
It edits. It's a
this is a procedural like a technique.
It edits the gene by precisely cutting the DNA and letting the DNA naturally repair itself. And take over. So the mutated DNA we would cut the DNA, and then the normal DNA would repair itself, and the mutated DNA would just go away, and it would repair itself. So the system consists of two parts: the enzyme called Cas9 and the guide RNA. The Cas9's function is it acts as a molecular scissors to cut the DNA. So this enzyme is what cuts the DNA at a specific location. And the guide RNA is the type of RNA molecule that binds to the Cas. So it's like a substrate acting to its active site within a cell. And it's based on the sequence, and it just helps the Cas.
Okay, this is very molecular biology, okay? Yes, okay. You very like the molecular biology?
Yes, I'm very interested in biology and chemistry in general, and I would like to be a doctor and hopefully specialize in a field regarding molecular medicine.
Molecular biology. It's very nice.